I'm actually the director of the software engineering group within the center. And so I've got a brief slide on the center in my background. So it's a multidisciplinary center, and we have 6 academic focus areas, and one of them is computational chemistry, and then there's also bioinformatics, which overlaps with this project, and 2 core capabilities: a high-performance computing center and a software engineering center. And I'm the director of the software engineering group in the center, and I work with collaborators to develop software for the funded projects that they have. And so, for example, the LIFE project that I'm going to talk about today, we've developed the software for that. But I've also worked on projects that are funded by NOAA, and we're doing like computer vision projects to classify and identify plankton in images and stuff like that. So I'm going to just start with the aims for the project. So, and then really quickly go over it. So it's the links. So LIFE, which is what we call our part of the project, stands for Links Information Framework. And these are the aims. I'm going to go back over the aims briefly in a minute. So the Links program is a broader project that is funded by NIH to generate basically a database of signatures, and then make those signatures available for research, and also to develop computational tools and approaches for analyzing those signatures. There are a bunch of centers involved in the Links project. There are 2 large data producing centers. One is at the Broad Institute, and the other is at the Harvard Medical School. And then there are a number of other centers that are developing technology, like new. These guys at the University of Arizona at Arizona State are doing some very interesting stuff. And so they're developing, laboratory technologies and things like that. And then there's us way down here, and I work with Stefan, who you may know, Stefan Schuer, and we have some funding to develop the LIFE framework that I'm going to talk about. The kinds of data that are being produced by the Links project are large-scale transcriptional profiling data, they're biochemical assay data, and a lot of different cell-based screening data. So the Broad produces this, and pretty much the rest of these are being produced right now by the HMS, the Harvard Medical School. So our near-term goals, what we're trying to do, is enable discovery of this data and provide a tool an application that will help people to go in and find from this data what things that they can use for their research, and for people who aren't necessarily familiar with the assays and how this data was generated. The main challenges here are that we're trying to address through here: modeling and representation, and building a tool for search, or more than one tool potentially. This URL is the URL to our project's website, the LIFE project website. And this is the system. So, or the actual, what we put in our proposal to build is a system like this. But really, just a little more simply, and this diagram got munched a little bit, is we want to just facilitate discovery of information from these links data producers, these computational centers who are producing tools that work with this data to analyze it, and also make that searchable using concepts that are familiar to a biologist. So a biologist could go in and say, I'm interested in all inhibitors of a particular pathway that don't inhibit a particular protein in that pathway. And then we try to give them those results that are from in the data set, and also maybe and ideally link with those results tools that can be used to analyze those results, or help them to analyze those results. These arrows don't really represent us holding this data. We don't really want to hold it. We just want to show people where it's at. And so we're going to do that. There's basically there's 3 steps for us doing it. So one is to develop standards, and also an additional ontology to help describe the data from these sources, and then build a semantically enabled software application, and to deliver that, and then enhance that data through some computational and some other techniques, so that that knowledge or that higher level information can be shown through the software. And so right now, and so in aim 1, I'm talking about that really means here the bioassay ontology, and then the life ontology, and some metadata standards. So I'm going to go through this ontology quickly. So we have, from an earlier project, we developed this ontology called the bioassay ontology, and what we mean by ontologies are controlled vocabularies, and then in our case also a more formal OWL-based ontology. Exactly, it's the same one. So Bard makes use of that as well, and the reason the ontology was developed is because there's a lot of different representations of this information. We want to come up with some sort of consensus representation that then we can use to search things with, at least from my perspective. So the BAO basically it was developed to describe assays, experimental results. And so there's two versions of the BAO right now, and the version 1 is currently, I think, in use, or is the Bard makes use of, and it describes things like assay format, design, the perturbagens, like the small molecules that are used in the assay, and how those things are described and pulled together. In the second version, BAO 2, information about biology was added to this ontology so that you can then, so it ties in this information of cell line information, tissue, even I think disease is on here, so that we can relate that to those experimental results. So you can search using a disease concept to find assay results related to that disease. And that was essential for us to do to develop the life ontology. And this slide just shows that the BAO uses a bunch of other ontologies, references a lot of other ontologies. So we don't, we use the gene ontology, for example, and use those terms. Those terms are referenced by the BAO. And these are, so the Bard also is using it. I'm personally not involved in the Bard project. Like we have multiple projects going on, but they're using it. We also use it in Life. The project I'm talking about now, we have another project called Region Base, which is also using it. Okay, so the links, the thing that we propose to do that we've more or less done is build a framework that will take data from these different link centers and then make them available to investigators, like I was saying, in a simple way. So this is so it's not just the ontology that we've built here. We built a bunch of software components. I'm going to describe a few of those, and then I'm going to show how this user interface works. So, and I'll go through this briefly. So one of the key things we had to do at the beginning is establish some metadata standards to describe things like, so that the centers could submit this data with, so that we know the field, they call a field, the field names that come in correspond to known concepts like, they're agreed upon, like cell lines, siRNAs, proteins, and just establishing these metadata standards took a little while in there, and it's an ongoing part of the Links project. There's a web page that goes along with it, but those metadata standards are used in the ontology. And so now the life ontology are used to actually describe the data that we put into the ontology. And so the life ontology contains elements from BAO that describe small molecule information, the specific links assays, so that we don't have to re-describe those. So a kinome scan assay is a kind of an assay that's already described in that we describe using the BAO concepts. And then we describe cell lines, diseases, genes, and include things like kinase domain classification, so you can query by group and family and things like that to find information. And then we also have, these are examples of things that are included in the ontology, and then references to the disease ontology, so you can query by disease and find things in there. So we also built this semantically enabled web app application to search it. So it's responsibly designed, and it facilitates search and exploration. And this is what it looks like. So it has this search box where you can just type in a search. You can also switch through different views of this. I'm very fond of this sunburst that shows the distribution of data in the entire links data set. And this is the architecture that we built it on. So it's a RESTful application, and we also use Solr, like they use in the Bard. We have a relational database. We have a triple store that stores the stuff that we've modeled, the information we've modeled using the OWL ontology. And we use, of course, JChem for structure-based searching. And so, and also for doing some analysis of the data. So I think I have here. So I'm going to show just a few of the features in here, and then I'm going to go through a couple of use cases in a little bit. So this is the front page again. There are different of these sunbursts, if you click on these, that show the distribution of data and based on these different views of it. So by protein, using the information in the protein ontology, or using, which is what this is showing, and then, or by diseases. If you type into the search box, it suggests terms using, this all comes out of Solr, and Solr is, if you haven't used it, it's an excellent open source tool for building nice search applications. So we suggest two kinds of things here. There's terms that are suggested. So those are just terms that we've indexed out of the whole data set, and they have they're not necessarily structured. They're not in an ontology. And if they show up under this concept bar, they match terms in the ontology that we have. And then if you mouse over that term and this suggests, it'll tell you, it brings this little pop-up and it shows you where in the ontology that falls. So, organ system benign neoplasm is in the disease ontology, and it says where that is. And that's useful because sometimes you'll type in a term like junk, JNK2, and you might want to search it might be referencing a protein, or it could be referencing a gene under this circumstance. You can see where it is. This is primarily how we show the results. So if you search for organ system benign neoplasm, we have categories of results are shown here. If you click on, so these show the number of results. So there are 42 compounds that match this query, 3 cell lines, and 3 assays. And by matching it means those things are joined together because they participated in some sort of experiment together. And so which means that these 42 compounds were run on 3 different kinds of, well, not necessarily all together, but there are 3 types of assays across 3 cell lines that involve 42 compounds involving neoplasms, or benign neoplasms. You can the different elements on here. If you click on to see details about, say, an individual compound, you get a page that shows structure, other attributes of the compound, and then you can browse experimental results and see some of the results and download results for individual compounds. You can also download results for your list of things that you have there. Of course, you can also do structure-based searching, and that's shown in the same kind of list, and it'll include, in this case, there were genes that were relevant. So these compounds were run on an assay where, basically in that, the L1000 assay, which is expression. And so we have different ways of filtering this data, rapidly filtering it, using tags that come out of the ontology, and I'll show you an example of that. And then we also have enhanced the information through some processing. So this is the basic pipeline that we use to bring the data in. And we're actually replacing the staging database. We use the staging database to sort of normalize the information or standardize it as we brought it in from these 2 centers. But now that we've established some more, the Broad has some APIs that we use now. And with the Harvard Medical School, we also established some APIs that we can call directly. So they're RESTful, they're JSON. We call and we get JSON back, and we can use that to ingest the data. into the database. Like I said, we put it into a triple store and also a relational database and enhance it through reasoning. And we also enhance this information using some other computational techniques, and that's not on this diagram. But the key thing that we have here that enables the search to work is we have a component that we call a journalist that assembles and presents the information to the indexer, so the Solr indexer, which is a text-based search engine. We present it, we basically create documents that describe the elements that we're trying to index. So in the primary index that we have, where we're indexing our perturbations, basically. So for each perturbation, we start to describe it. We say this gene is involved in this, this protein, and the synonyms, these are the synonyms for this protein, and things like that. And all of that ends up in a document that gets thrown into the indexer. Okay, so this is an example of, then on top of that, how we've added just a little bit more information on top so that you can search. We did some analysis to identify, say, promiscuous inhibitors in the data set. So as we load the data, we also did some analysis, we did various kinds of analysis. Just one example is promiscuous kinase inhibitors. So, and as I said, we also add on top of there, pull in concepts from other ontologies. So for example, the cell lines have an associated disease, and we tie that to the disease ontology, which allows us to do subsumption queries, which I'll also give an example of. So in here, what this is showing, the sunburst shows, if you click on the small molecules, are that there are 21 inhibitors we identified in the data set that are promiscuous kinase inhibitors. And if you search that, you can search it here, and you'll get back those 21 promiscuous kinase inhibitors, the assays they've been run on, and what's involved in there, in what other experiments they've been involved in. And so this is just showing also the value of this text search. So now I've just filtered, I've searched for the promiscuous kinase inhibitors, and I'm only interested in ones that are active against JNK3. So I type in JNK3 and active, and it reduces the set from 21 small molecules to 7. And then if I further then open these tags, which are based on the ontology, and select, I think, PDB ligand, which appears here, now I've basically found all the PDB ligand promiscuous kinase inhibitors for JNK3. And then if I look at the data, this visualization that we put in here it's a visualization of all the kinases that we have by group and family, and then they're grouped in there. And blue is are inactives, and red is active. So you can see there's a lot of red for this particular kinase. And you can drill down, click on a family and see the detail, the results in a little more detail there. And so this is just to show the same thing for group selectivity. So we have group selective inhibitors we identified, and that's what a group selective inhibitor would look like in that same graphic. And you can drill down and see it in more detail. And then for subsumption, just to give a quick example, so we have this browse tree available from this browse menu here, and it shows how we've grouped the protein kinase domains, or how we're using those. And so this map-k maps to this map-k. If you mouse over that in the view, it shows you, and then it also shows it here in the suggest. And so if I query by map kinase, it only brings back those kinase proteins that are in that family. And then you can do additional manipulations with that and download the data. And this shows that the same thing works for diseases. So querying by benign neoplasm, it's a little easier to see it here, but what this shows is, so I queried by benign neoplasm, I've got 4 cell lines that came back. Only one of them says benign neoplasm. The others have these other diseases listed because they're all subsumed by benign neoplasm. So that's the idea of subsumption. I type in a higher level concept, and I can find all the information that's contained within that concept. And there are more, just to make this clear, in the disease ontology, there's a lot more things under benign neoplasm than are shown here, but we only include the piece in the application where there's data associated with it, because it would make no sense to show an entire tree of concepts and you click on them and get no results for everything all the time, right? A few other features, we can create lists, we can filter based on lists, you can download the data, and I showed you the keyword filtering. The lists are kind of handy. So we can go back to the group selective. So here's my 3 group selective compounds, and then if you click on these buttons, I can create a list of those compounds, and then I can go back and find, I'm going to search for all, lung carcinomas. And if I initially search that, I'll get 69 compounds that were screened against those. But I would just like to see the ones that were in my list that I created based on my earlier query. I can select those on the list, and I've reduced it. So only 2 of those compounds are run on those cell lines, and I can then download those results as well. We, of course, use Marvin for JScript to do structure-based searching, and this is just showing that feature. And I would say that we like this a lot better than, we like the Java app too, but this is much better. I like that. And so this is just showing that we can do structure-based search of the same data set, get this information back, and then I can filter it. So I'm interested in, I've got the results for these 2 molecules, but now for whatever reason, I'm only interested in results related to women. So I've filtered it on women, and if I look at the cell lines, the tissue type, the gender that those cell lines came from, that this molecule screened against are women. Okay, so the next steps so we have basically what we call an alpha release. It's publicly available. I'll show you the URL in a little bit. We're going to have a beta release before December where we're going to we'll have, our pipeline right now is, it's manually fired off, and we're going to get it in like an automated, it'll keep it regular. The goal is to keep it regularly updated, the index, and that's what we're planning to put in place before December. And then around the same time frame, we're going to integrate it with the linksproject.org search page, which is part of this Data Coordination Center pilot. So on the links project site, there's a search page, or search box, which currently returns search results like this. And so we're going to hook up, this is just a mock-up, but when you search in here, it'll bring back searches of results for the current website, but if you click here, then you can see the same results against the actual links data set. And this isn't going to say LIFEwrx, I just put this here. It's going to say data, is what it'll say. So if you click on data, it'll give you a distribution of the data in the data set, and if you click through from here, you'll be able to get back to the search application. And that's what we'd like to develop. So these are the URLs. This is the bioassay ontology URL, the life knowledge base URL, and this is, if you'd like to go check out the current alpha version of the application, you can go to that link, life.ccs.miami.edu. And these are all the people involved in the project. Here's Stefan, if you miss him, and all of the other people. So if you have any questions.
How about the Sunburst stuff? What is that coming from? Software package, and what kind of?
So the Sunburst comes out of a JavaScript library called D3JS. It's very popular. Lots of people use it. Okay. And so we modified. I don't know if you're looking for ways to visualize data. D3JS has got lots of great stuff, and so we pulled some stuff out of there and tweaked it slightly to do that. And we also, that other Sunburst, the one I showed for the kinome data, is nice because it's zoomable. You can click on it and it'll zoom into the different parts of it. And that also came from there.